easier to grab one of the handouts at the beginning. If you've got one, use it. If you don't have one, you can use what's on the screen. Here's what I need you to do. I would like you to read that paragraph. Take about 30 seconds. And then I'm going to ask a few people in the room to tell me the thing that stood out the most to you in that paragraph. One thing that stands out to you in that paragraph. 30 seconds starting now. Go. Ten seconds. Okay. I'm going to take one person from each side. I know a few names in here, but not very many. So I'm just going to pick on a couple people. Doug, what stood out to you?
The enormity of the task.
The enormity of the task. Okay, let's come over to this side. Yes, I see that raised hand, thank you.
Yeah, for me it's the 250,000 neurons being produced every minute.
Okay, so that stood out to a lot of people. 250,000 neurons per minute. Or per.
But technically enormity means wickedness. You're thinking of enormousness.
So, thank you. So, the 250,000 neurons per minute, that's a big one. Now, of course, that's time averaged over the whole course of development. There are fewer at the beginning. There are none at the beginning. And there are many, many more at the end. All right, that's all very impressive. Thank you for pointing those things out. I would like to point out what I think is the most amazing thing in this paragraph. I would like you to direct your attention towards that period at the end. Look on your paper. Look at that period. Look very closely. That is a 6-point Times New Roman period. And a 6-point Times New Roman period, it is exactly 100 microns in diameter. That's 100 millionths of a meter in diameter. We measured it on our microscope, so it's legit. That is our scale bar, 100 micron. That is what the size of that period is. That is, to me, the most amazing thing on that paragraph, because this is how you started your life, as a single fertilized zygote. That was 100 microns in diameter. So, take a look at the person next to you. That person started out the size of that period on that page. It's not much of an exaggeration to say that the process of how this speck, this period-sized cell, develops into you is the greatest show on Earth, and is our topic tonight, developmental biology. So a little bit about myself and my background. My name is Dr. Brown. I'm blessed with 8 beautiful children and my wife, Erin. They are not here tonight. You would probably know if they were. But we recently moved to Arizona. Last year we moved to Arizona, where I became the director of the Creation Research Society. Research is our middle name. So what is the Creation Research Society? It's the largest and oldest network of creation scientists in the world. It's founded in 1963. Henry Morris was one of the founding members of the Creation Research Society, and the whole purpose of the society is to do 4 things. We publish research. So I saw some of these Creation Research Society quarterlies out there on that back table. That's a publication of the Creation Research Society. So it's a peer-reviewed scientific publication around creation science. We present research. So we just came actually from our annual conference in St. Louis. We have it at different locations each year. And this is a technical conference. So it's not just a general conference about Adam and Eve, etc. This is a technical conference where you get into the genetics of Adam and Eve and things like that. So we had a great time. Good to see some people here tonight that were at that conference a week and a half ago. We fund research. So if you have the latest, greatest creation idea, or you think you do, you can submit for a grant, and we'll give you some seed money to pursue that idea, if it's a good one. And last but not least, we perform research. So this is why we moved to Phoenix, Arizona. We have a lab there on the campus of Arizona Christian University. Here's our team that are blessed to be able to work with undergraduates from the university, and it makes it fun, and they do all the grunt work in the lab. So, our lab does two things. We do molecular paleontology. So paleontology is the study of the dead remains, fossils in the earth. We do molecular paleontology, so we study the molecules that are in those dead remains, the biological molecules. The other thing that we do is something called phylogenetics, studying the genetic history of species and how they developed from the flood. But I'm not going to talk about any of those tonight. I'm going to talk about the greatest show on earth, developmental biology, which was what I did my PhD in. I am going to put this slide up at the end. Creation Research Society, we're creationists, so we don't receive government funds to do research. We rely on God's people to help support and fund the research. If you are a degreed scientist, you have a graduate degree or above in a scientific field, you need to be a member of the Creation Research Society. That's why we exist, to connect. We are stronger together if we're working together. So I'm going to put this up. If you just are interested in kind of following the current research, sign up for our newsletter. If you're a scientist, you should join. Okay, this is our motivation as a Bible-believing scientist. Scientists in general are just studying how the world works. The difference between a secular scientist and a Bible-believing scientist is that we want to give credit to whom credit is due. Thou art worthy, O Lord, to receive glory and honor and power, for thou hast created all things, and for thy pleasure they are and were created. That's why we do what we do. So I started down the road into science as a young man. This was at the University of West Florida. I earned my master's degree in genetics. Here, these tubes are filled with fruit flies. So we worked with fruit flies. We studied Alzheimer's disease in fruit flies. Now, fruit flies don't get Alzheimer's disease, but they have some of the same proteins, such as these guys right here, the gamma secretase complex, which in humans results in Alzheimer's disease. And so you can study those proteins, how they work in a fruit fly. But most importantly, what I learned during my master's degree, I learned about God's favorite science. Did you know God has a favorite field of science? This was just staring me in the face all of these years, and I can't believe I missed it. And then one day I was looking at the first book of the Bible, and there it was, God's favorite science, the first 5 letters, genes. Genes. And ever since then, I have followed and pursued this field of science called genetics. And that's what we'll be talking about tonight. We're not talking about just how things develop, but how they develop at the molecular level. So I went on to get my PhD in molecular biology and genetics at Cornell University. That's also known as tiny stuff, which you cannot see without a very expensive microscope. That's some of the tiny stuff we're going to be talking about tonight. Here is, in a nutshell, what I was studying. So this is that cell that I showed you at the beginning. The single cell that we all started at, called a zygote. And that cell undergoes a process known as developmental biology, or development. And it goes from a single cell to a ball of cells, to a hollow sphere of cells, to an embryo, eventually to a mouse, in the case of what we were studying. So we studied this in mice. Let me show you an example of a couple of the embryos that I worked with. I'm going to try to make you into developmental biologists tonight. So let's start here. I have two embryos on the screen. These are X-ray computed tomography images at the micro scale. I want you to take a good long look at these, and then we're going to vote. One of them is a normal mouse embryo, 10.5 days through development. One of them is a mutant mouse embryo, about 10.5 days through development. Well, don't, Warren, don't be blurting out answers yet. Okay, we're going to vote. By a show of hands, how many would say the one on the left is messed up? By a show of hands, how many would say the one on the right is messed up? Okay, it's about 50-50. It's always about 50-50, which is interesting. All right, so we're about to separate the A's from the B's. The mutant embryo is the one on the left. And let me tell you why. Here in the normal mouse embryo, we have along the back of the embryo this structure called the neural tube. Up here, that neural tube will develop into the brain. On the rest of it, it'll develop into the spinal cord. Now, perhaps you noticed as it was rotating, spinning around, that this one has a neural tube which is open. So the neural tube starts as a flat plate and it curls up into a hollow tube. If that process doesn't happen properly, it remains open, then it causes neurodegeneration, and this is a birth defect called exencephaly. This happens in humans as well, which is why we study it in the mouse. Another form of this, which you may have heard of, is called spina bifida. That's when it doesn't close properly in the spinal region. So this is in the cranial region, the brain. All right, so when I think about developmental biology and think about the complexity of this process, think about what has to happen. You have to go from one cell, which divides into 2 cells, which divides into 4 cells, 8 cells, 16 cells. And somewhere along the way, those cells have to be told, okay, you cells become an arm. You cells, I want you to start beating rhythmically, contracting. You're the heart. You cells, I want you to start sending electrical impulses. You're a nerve. And that all has to be programmed into that first cell. I'd like to make this comparison to a Lego. Humans develop and build and design some pretty incredible things, including some pretty incredible Lego sets. So, does anybody here know, you guys look like avid Lego builders, does anybody here know what the most expensive Lego set is? Any guesses? Well, okay, and I'm not actually asking, I'll tell you the price. It's not that much. The most expensive Lego set is Star Wars Millennium Falcon. And here's why. It's because they're made up of 7,541 pieces, 8 minifigs, a 495-page instruction manual, and all this for the low, low price of $850. It's not so bad. And once you go through all this work of assembling it, you have a beautiful Millennium Falcon. Now that's a pretty nice design, isn't it? Like, it's pretty impressive. Humans are able to design and build some pretty impressive things, but there is a key fundamental difference between this and development. For instance, let's look at one of these impressive creatures from God's creation. We have the ruby-throated hummingbird, native to Illinois. Their heart beats up to 1,220 beats per minute. That's impressive. Figure 8 wing beat pattern, so they can generate lift in the downstroke and the upstroke. Amazing design. Those wings beat 46 beats per second, 250 breaths per minute. This is an absolute marvel of engineering. But you know what I think is the most incredible thing? This entire machine is completely self-assembled from the instructions contained in this tiny package, the hummingbird egg, that single first cell. Let's look at another example, the Venus flytrap, another incredible machine, a marvel of engineering. This trap has these little trigger hairs that are set off when an insect visits. It's got these cells that secrete digestive juices to digest that hapless insect. They secrete their own bait, so they have cells that secrete the sugary like nectar that attracts the insects. They even have these bars to hold the insect in after they've got it. It's an amazingly engineered system. But you know what I think is the most incredible thing about this is that it is entirely self-assembled from the instructions contained in this tiny package, the seed from the Venus flytrap. So, as impressive as Legos are, or Lego are, I would truly be impressed when they develop a Lego set that contains a single Lego. And all you have to do is set that Lego there, and it will completely divide and self-assemble into the finished Millennium Falcon. And you can begin to appreciate, as impressive as human engineering is, and human design is, and it is impressive, we are nowhere near the level of design to create these living organisms. Now, how do things develop from that single cell to a full-grown organism? This has been a mystery for millennia. And you see glimpses of this mystery even in the scriptures. As thou knowest not, the wisdom of Solomon speaking here, thou knowest not what is the way of the Spirit, nor how the bones do grow in the womb of her that is with child. It's a mystery. In fact, the early ideas on development believed it must have happened something like this. So here was the idea that inside of each of the sperm cells was a teeny tiny fully formed human packed in there. And then that human just got larger during development. And can you blame them? It seems silly now, but can you blame them for thinking that? Like, how else would this come about? So those were the early ideas. We know a lot more now. Here's an example of what we know now. So this is a video of the development from a single cell to a larval stage of the alpine newt. So we've learned an awful lot about how things are happening from a single cell to a full-grown organism. But how do you go about studying and learning about human development? You can do it in like a frog or a salamander pretty easily. They develop externally. How do you do it in humans? That's tricky. Well, here's two ways that you do it. Number one, you use a model organism, such as a mouse. So let me leave this with you just as a freebie. Why would God create other organisms to be so similar to humans? Couldn't he have created us uniquely different from everything? He could have, and that would have been a poor design, because then you wouldn't have been able to learn about your own biology unless you studied it in humans. But because he created us similar to these biological organisms, we can study a mouse and learn about human development. So that's one thing we do. We've got the mouse embryo over here on the left, the human embryo on the right, at 28 days, 9.5 days old. A lot of the same structures. There's the neural tube, there's the neural tube. There's the eyeball, there's the eyeball. There's the lower jaw, there's the lower jaw. There's the heart, there's the heart. There's the limb bud, there's the limb bud, the beginning of the limbs. So you can study these things in a mouse. Okay, can you tell which one's the human and which one is the... So they do look a little different on the end of the process. In case you weren't sure, this is the mouse over here on the left. Development lasts 20 days in a mouse, so it's very rapid. I'll say a little bit more about that later. 266 days approximately in a human. This is my 8th child, our daughter Genesis is her name. So that's one way you can study development, is by using a model organism. Here's another way that you can study development. We asked you this question at the beginning, We have a messed up embryo on the left, a normal mouse embryo on the right. You can study abnormal development to understand normal development. So you study development when it goes wrong, and that tells you how it should be going right. I'll explain. So here's what we did in my lab at Cornell University during my graduate studies. We would take a mouse and we would inject it with a chemical, a pretty nasty chemical, and that would cause birth defects. And then we would isolate those birth defects, and we would try to figure out which gene has a mutation in it that's causing that birth defect. Once you identify the gene, then you know this gene, gene A, is responsible for this developmental process. You can see how that logic works. Here's what the experiment actually looks like. So we would take a mouse, we would start with this black mouse, and this would be a male. And then we would inject this thing called ethyl nitrosourea. It's a pretty harsh mutagen, and it causes mutations throughout the mouse genome. Those mutations are represented by those stars. So I've got a red star up there and a yellow star up there. Then we would take this mouse, we would cross it to a healthy female, and we would use a white mouse. There's a reason we use these different strains of mice, and I'm not going to explain the details behind that, but there is a method to the madness. So in the offspring would randomly receive a subset of the original mutations from the dad. So you see how this one got the yellow mutation, this one got the red mutation. That's the idea. We would then take those offspring and we would cross them again to a white mouse. In those offspring, this red star, the red mutation, is going to be inherited by half of the children. Now, we can't see the mutations, so this is all blind. We just know probability half of the offspring will have the mutation. Okay, so then we take these offspring and we cross them back to their dad. You're allowed to do that in mice. So when you cross them back to their dad, half of those crosses will be a mutant versus a mutant, and you will get a ratio of embryos that are carrying double mutations, and that spells bad news for that embryo. And that's what we're looking for. We're looking for those double mutations. So we would look at in the embryonic stages of development to identify birth defects. Now this involved a lot of mice, as you can imagine. This was the injection stage. All of those like shoebox-sized cages in there, those are cages for mice. And then you get into breeding. Those crosses I just showed you, and more breeding and more breeding. So each of those cages is filled with 5 to 8 mice. And so this is a team effort. It required a lot of people. These are not creation scientists or even Christians, but here's an important concept. All empirical science is creation science. Just don't tell them that. Okay? Because if God made the world, science is just the study of how the world works. So anytime an atheist is studying how the world works, guess what? They are doing creation science. Our team here was studying how this worked, and here were some of the things we were looking for in the embryos. We were looking at these pharyngeal arches. They form the throat structures. We were looking at the heart, looking for heart defects, the brain, the neural tube, craniofacial things like cleft palate in the embryos. We were looking at the limb buds, counting the digits, making sure they had all 5 digits. We are also looking at the extra embryonic membranes like the placenta, which is not shown here. Here are the mutants that came out of this screen. That's what you call this type of experiment. It's a screen. Here's a list of all these different mutants. We had some that had these craniofacial defects, so stuff wrong with their heads. We had others that had these defects in the extra embryonic membranes. This is the placenta from one of these embryos. We had these that had heart defects, and others that had these kind of nondescript defects that we couldn't really group into any other group. Now, once you have a defect, that's only half the story. You now have to find, out of the 3 billion letters in the mouse genome, 3 billion with a B, one of those letters has been changed that is causing that birth defect. 1 out of the 3 billion, and you have to go find it. And it's a pretty lengthy process, and we won't talk about it, but this is what we're looking for. That's what I mean by finding the needle in the haystack. We had to go and find that mutation, the letter that was changed that was causing the birth defect. So I'm going to use these embryos to tell you 2 stories about development. There's a lot that happens in development, and we're just going to look at 2 of those stories. Part 1, choosing sides. I'm going to use this mutant right here. It was named 13B. They get boring serialized names. This was 13B. So take a good look at 13B. I'm going to do the same thing. I'm going to rotate it around in this micro CT scan, and I want you to look for things that are wrong in the mutant, which is on the right side, compared to the wild type. And we'll take some answers from the audience. You're looking for things that are wrong in the mutant compared to the wild type. Okay, we're going to go back and forth. I'm going to take raised hands, 1 from each side. Let's start over here. Raised hand, something that you see that's wrong in the mutant. Go ahead.
Well, the spinal column again,
And what about it?
it doesn't look complete for one thing.
Okay, yeah. So you notice like it's still open down here, and you notice how it's kind of spiraling. Good observation. Let's go over here. Something that's wrong in the mutant compared to the wild type. Go ahead.
It looks like the spine divides differently.
And are you talking about like up here at the top?
On the backside. Coming around.
Oh, coming around. So clearly there is something going on with that spine, because up here at the top as well, and in here, it's closed incorrectly. Okay, let's do one more over here.
Prominent ridge in the front of the face.
Say again?
A prominent ridge in the front of the face.
Prominent ridge in the front of the face. Let's let it come around. Right here.
Like a smart view.
Yep. Okay, let me point out a couple other things. Did you notice it has this giant, almost puffy cheek-looking thing? Well, that's interesting. That's the maxillary process. That's the upper jaw, or it will become the upper jaw in the embryo, and it's enlarged. But there's something very unusual about this embryo that nobody has mentioned yet, and that's because it's very hard to see first glance. Here's a wild type embryo, and I want you to look at the heart. The heart in the wild type embryo, it rotates counterclockwise. You see the arrow? Okay, let's look at the same orientation in the mutant. You see how it rotates clockwise? It's reversed. So the left and right sides are reversed in this mutant. Well, what am I talking about reversed? I'm so glad you asked. If you took this circle and you cut it in half, are both halves the same? Yeah, that's called bilateral symmetry, where both halves are identical. What if you took this star and you cut it in half? Are both halves the same? Yep, the star also has bilateral symmetry. What if you took a human and you cut the human in half? Are both sides the same? Now on the outside it sort of looks like we have bilateral symmetry, but on the inside it's far from being the same. Here is why. You've got your heart that's a little more tilted onto the left side, you've got your stomach more on the right side, you've got the liver more on the left side, or on the right side. So it is not the same on the inside. And in fact, if something goes wrong during development, it can be completely inverted. This is actually a condition known as situs inversus, where your internal organs are all a mirror image of what everybody else has. Now, if it's a clean swap, if it's a perfect inversion, you would live your entire life and not even know. The only difference you would notice was that the doctor would have a hard time finding a heartbeat on your left side of your chest, because they would have to look on the right side. Here's a fellow from the UK. He lived 92 years with backwards organs. Full situs inversus, full inversion, and he was fine with it. The problem arises when it doesn't happen quite right, when it can't choose sides. That's what I mean by choosing sides. So here's how we started to look at this. You are looking at an embryo head on there. This is a normal wild type embryo. It doesn't even look like anything. It looks like a blob of tissue, doesn't it? Well, that is an early mouse embryo. And you notice we are detecting a gene called lefty, and it's marked by this purple color. And this is correctly labeled left on the right-hand side of the screen. And that is where lefty is expressed, just on the left-hand side of the embryo. There you can see it in a cross-section. Now let's see where lefty is expressed in the mutant. Doing the same thing, do you see what happened? It's expressed on both sides. In other words, this mutant can't decide which side is left and which side is right. It's confused. So what could be causing this mix-up? I'm so glad you asked. So we had to go find the needle in the haystack, find the gene that was broken that was causing this defect, and here's the gene that was causing the defect, a nonsense mutation in PIBF1. That's the gene, also known as the progesterone immunomodulatory binding factor 1. Doesn't that sound exciting? So what in the world does this immunomodulatory binding factor 1 have to do with choosing sides? So we started looking a little closer. And this is scanning an electron microscopy of the brain. Here is a normal mouse. If you were to zoom in, high power magnification on the cells in the brain, you would see these little structures. One of them, and exactly one of them on every single cell. This is called, and by the way, there is one of them on every single cell in your body. These are these special little antennae that grow on each cell, one per cell. They're made for sending and receiving signals and some other special functions, and they're called primary cilium. I bet you didn't even know you had them. If you did not have them, you would not be sitting here today. So we looked at the mutant, and you zoom, and they're all gone. There are none of them there. Here is a cross-section. If you took this embryo, put it through a deli slicer, and looked at the thin cross-section of it, these are all of the red is marking all of those cilia, one per cell in the brain. And in the mutant, there are none. There's just this background staining. So all of the primary cilia were gone in this mutant. Wait a second. So primary cilia are influencing the left-right decision. How in the world does that happen? Well, the cilia are missing everywhere, including in the very early embryo. If you were to look in a very early embryo, this is about 7 days into development. We're looking down at it as if this little spot there is kind of like its belly button. That's not quite accurate, but you can think of it, that's kind of like its belly button. And if you were to look into this structure called the node, you would find all of these primary cilia. That's what those little stringy things are. If we look in the same place in the mutant, they're gone. There are none. What does this have to do with choosing sides? Here's what it has to do with choosing sides. So that tiny little belly button structure called the node with these tiny little primary cilia, early on in development, all of those cilia in the node start to move. And they all start spinning in the same direction. When you start spinning in the same direction, what does that create? It creates a flow of molecules in one direction but not the other. And it sends a signal over to the left side of the embryo, and it tells the left side to become the left side. If you remove those, it doesn't know which side is which, all because of those tiny little cilia. This is a paper that published a video of these little cilia spinning in the node. That's what they look like. Here's the incredible thing. How many genes do you think are involved in making these primary cilia? Let's look. So it's a pretty involved process. It involves many steps. Here's step number 1, the formation of this mother centriole and the assembly of what's called a basal body. These are all the genes and proteins that are involved in that step. There's the one that we were studying, just one of them. And then, of course, those vesicles have to start growing, and you get this ciliary vesicle. Those are all the genes involved in that. And then it has to elongate and get even further. These are all the genes that are involved in that elongation step. And then it has to dock on the membrane of the cell. These are all the genes that are involved in that docking process. And then it has to function properly. So it has to work as an antenna, sending and receiving signals. These are all the genes that are sending and receiving signals. And I'm going to be quizzing you on this at the end. In short, 686 genes are involved in cilia formation and function. 686. And if you remove just one of these, the whole system fails. Not just the cilia formation. Your development fails by just removing one. I will praise thee, for I am fearfully and wonderfully made. Marvelous are thy works, and that my soul knoweth right well. Let's look at one more story, shall we? I'm going to tell you a story about this guy right here, 1D. Part 2, thinking like a burrito. So I showed you this guy earlier. He has an open neural tube. The neural tube doesn't close properly. Here's how it's supposed to close. This is day 8 during mouse development. This is day 9.5. Open, closed. Here's what it looks like between that time period. Those folds gradually come together until you eventually close that tube. Here's what it looks, by the way, this is hard to see in a mouse because a mouse develops in utero. But the technology now exists to be able to extract those embryos and grow them artificially and image them. So this is some of the first imaging of this process. We're looking from a top-down view. These are the neural folds. They're forming a valley. They're coming together. And there's the tube, like a zipper in each direction, closing up. And you have your early brain because of that process. You know what I see? I see a burrito that's about to learn how to think. So what controls this intricate folding? A lot of things do. And clearly, one of those things was messed up in our mutant. One of the genes that controls this was messed up in our mutant. So we went and found the needle in the haystack. And here's the needle in the haystack. This is the wild type, the normal sequence, and it has the letter T there in its DNA. The mutant has a letter A there instead of a T. One letter change out of 3 billion with a B, and the whole system fails. Okay, so what actually was changing? That letter change impacted this protein. Do you see that little red blob in there? That's one amino acid that was changed in this protein, and the whole system failed. What is SPCA1? Oh, that's the secretory pathway calcium ATPase1. Secretory pathway calcium ATPase1, it's a pump that moves calcium around the cell. Specifically, it lives over here on this area of the cell called the Golgi apparatus. This is the sending, receiving portion of the cell, the UPS center of the cell. SPCA1 lives on the Golgi, and it pumps calcium from the interior of the cell to the interior of the Golgi. That's its function. What in the world does moving calcium have to do with neural tube closure? So we tried this experiment. It was one of my favorite experiments we did. We wanted to see if calcium was changing during neural tube closure. So this is hard to see in mouse. Let me walk you through this experiment in a chicken. Why chicken? All you have to do is open up the egg and voila. It's right there in front of you developing. So we took this chicken egg, you open it up during development, and we would inject it with this DNA. The DNA contained a special protein, this guy right here. This protein glows green, a fluorescent green color, but it glows brighter when there's a lot of calcium, and it's dimmer when there's not a lot of calcium. So we can use it to see what the calcium levels are. So we then shock the embryo. This causes the cells to take the DNA in. We fast forward a little bit and we image the live neural tube closures and see what's happening with the calcium. So I'm going to show you 2 short little videos. This is the chicken embryo. Here's the head region with the neural folds closing. This is the tail region. I'm going to show you a video from each of these. So here's the head region. Each of these blobs, the green blobs, is a cell. And the green represents the calcium. Changes in the green represents changes in the calcium levels. I want you to see if you notice anything on this video. Watch closely, because it's pretty subtle. What did you notice was happening to individual cells throughout that video? Who said twinkle? That's exactly the word I had in mind when I saw this for the first time. It looks like a twinkling. What that twinkling is, it's calcium coming in and going out, coming in and going out, pumping in, pumping out. Okay, let's look at the tail region where the spinal cord is closing. Same thing. Who said thunderstorm? You, sir, are a developmental biologist. That's exactly what I thought. It looks like a lightning storm with these flashes, so it's happening in groups of cells. A flood of calcium in, and then it pulls it out. A flood of calcium in and pulls it out. What in the world is all this calcium doing, flowing in and out of these cells? Well, we don't know the whole story. There's still more questions to answer. But what's essentially happening is that neural tube is starting out flat. The calcium going in and out is causing it to constrict. And when it constricts, it folds. And you have a brain because of that. If you want to read the whole paper, secretory pathway calcium ATPase1 controls mouse neural tube closure by regulating cytoskeletal dynamics. It's good reading material at bedtime. It'll put you right to sleep. So here's what I like to think about when I see this sort of system. Raise your hand if you've heard of the idea of irreducible complexity. Good, that's most of you. You are looking at a highly irreducibly complex system. Let me review what that means. Irreducible complexity basically means the whole is greater than the sum of the parts. In other words, here's a Boeing 747, which can fly, and it's made up of 600 million parts which can't fly. Think about that the next time you're flying in an airplane. Just don't think about it too hard. That's irreducible complexity. The whole is greater than the sum of the parts. Okay, so do you see some irreducible complexity here? Here is a single amino acid, a protein that's composed of 952 amino acids. If you remove just one of these amino acids, if you change it, that pump no longer works. This is an irreducibly complex protein. But this irreducibly complex protein is part of an irreducibly complex mechanism called neural tube closure, because it's not just one gene that's involved. 240 genes and proteins are involved in closing this tube. And we messed up one of those, and the system failed. So we have layers of irreducible complexity in this system. You know what I see? I see double-decker irreducible complexity. But it doesn't end there. We have an irreducibly complex protein, which is part of an irreducibly complex mechanism, neural tube closure, which is part of an irreducibly complex system. The brain is just one of the things that has to develop properly for this organism to develop. All of the rest of it also has to function. Do you know what I see here? Triple-decker irreducible complexity, layers of irreducible complexity. I will praise thee, for I am fearfully and wonderfully made. Marvelous are thy works. You add to this the brain, the eye, the heart, the rest of the vertebral column, the limbs, facial structures. You put all this together, and it is no exaggeration to say that the development from a single cell to a full-grown organism is the greatest show on earth. Now, we've looked at a lot of trees tonight, a lot of details. I don't want to lose sight of the forest for the trees. So I like to end by looking at the forest. Very good indeed. And let's take some questions.